It's okay. I don't have that effect anymore. Let us begin. We'll pick up where we left off last time. And we left off at protein. Protein. So I sent you, I did it out of sequence. I really think I have to, proteins and nucleic acids have to be explained fairly thoroughly, and it lends itself to my terrible demonstrations and my trying to give you an idea of what's going on, I think. And this is the hard part. It's all about shape, particularly proteins. You can see, you get them. Everything we're starting to see is the relationship to shape, and there's nothing more demonstrative about that, or demonstrable, way to say it, that demonstrates it better than proteins. And so we're going to, and effectively, and I say it all the time, you are what your proteins say you are. The next unit after that is DNA and RNA. The whole idea of what DNA does two things. It replicates so we can make more cells, and it tells the cells basically what proteins to make and ergo how to behave. Everybody in this room is about 98% identical DNA. There's a couple of percent of DNA that we don't exactly, so we're all the same. It just drives me crazy. There are subtle differences between us that are genetically oriented, and I mean, whatever, whether you're tall, whether you're short, whether you're athletic, whether you're coordinated, or if you're like me, you're a klutz. I mean, figure it all out. But all that is some subtle changes genetically speaking, and I really think we should look more at our similarities than our differences. But that's a whole that's a lecture for a different discipline, not science and biology. But nevertheless, proteins. And so they're so vitally important, and it's all about their shape. Their functions are dramatically different. Structural proteins in and of themselves you already learned about, but I'll tell you why they are so structurally sound when we look at the structure. Collagen, elastin, reticulin. Ones that you're going to learn about, we have a whole unit dedicated to it when we start looking at skeletal muscle. Our third unit, that's actin, and particularly myosin, which is a protein that actually changes shape to create movement. So these are, we call them motor proteins, as you will see. So they create movement. And we address enzymes in today's presentation. Every chemical reaction, and I can't even think of one off the top of my head that does so without, basically revolves around the availability of enzymes. Very, very complicated, large protein structures that take that in a matter, and it's not hyperbole, it's not overblowing how important they are, but in a minute you can have millions of reactions that this protein helps to make occur very, very quickly without being used up, and genetically determines what proteins, particularly what enzymes. I use the difference. The difference between, it's a sad difference, but if you study genetics, it's called autosomal recessive and autosomal dominant. You hear about these different genetic things. I don't know whether you studied it or you will, but one of the things that you can, if you elect to see if you're going to have kids, is are there going to be, 50 years ago, there were hardly any tests. My first wife and I tested in 1980 at McGee, where it was the earliest days for genetic counseling, for a disease called Tay-Sachs disease, which is prevalence the wrong term, but it's most likely found in people of our extraction, which are Eastern European Jews called Ashkenazi Jews. And if you are a carrier, and you and your partner are carriers, there's a 1 in 4 chance. That's what genetics is about. Looking at the odds of something happening, that good or bad. 1 in 4 chance that the baby, doesn't matter whether it's male or female, will be born with Tay-Sachs disease. The tragedy is it affects the neurologic system. It's an enzyme that allows the waste products from assembling part of the nervous system, a protein and fatty compound, from assembling this to get rid of those waste products. If you can't get rid of the garbage, the waste products, they accumulate and they cause damage. It's a fancy term called the lysosomal storage disease. We'll revisit it again when we look at lysosomes, when we look at the cell. And what happens then, that baby develops normally for the first year and a half, all those wonderful landmarks, crawling and head up and walking and talking. And after that, it goes downward hill, and the baby just loses all control and eventually does not survive past age 5. Normal baby for a year, year and a half, everybody's happy, no warning, nothing you can do about it. Terrible disease. A lot of reasons why people may or may not choose to have children biologically, or that's something that they wish to do if conceived a child. Maybe you like to terminate the pregnancy. That's a whole other sub-issue for, of course, not named biology and biological science. But nevertheless, you get the idea. That's how important these darn things are. And yes, it's about their shape that allows them to make these things available to break down those products so that your body can reliably excrete them. You're going to see movement for creating for muscles. Before, when we looked at carbohydrates and lipids, okay, they had a lot of carbon, hydrogen, and oxygen. Oxygen more so in carbs, less so in lipids. Now we're going to add nitrogen to the party, as you will see shortly. The poster child for all of this is a unique molecule that's both acid and alkaline. It's called an amino acid. You'll see a better diagram than I will write, but it has a central carbon, a hydrogen, something called an R group. That's the barrier. There's up to 20 different of these R's that we use. They're all different materials. It could be as simple as a hydrogen or as complex as a relatively large molecule. On one side, it has an acid, that C double bond OH, and on the other side, it has an NH2, we call an amino group. Interestingly, that's basic chemically. This is acid chemically. So they neutralize each other. It's a neutral molecule. That's the building block. They're all made of these chains together. Effectively, they're identical units that vary by the R group. And the R group, as you will see, have components that can make them either acidic or basic, positive or negative, water soluble or fat soluble. All what the R groups are. 8 of these amino acids, some authors say 9. I won't ask you to check 8 or 9, which one is positive. I'll say 8 or 9, or just put one or the other, are considered essential. Essential, we talked about this, essential amino acids, yes. Yes, we did. Yeah, so you know what I'm talking about. The whole tofu thing. Yeah. You got to acquire them by diet. Somebody's paying attention, not me. I got to talk to 3 different classes, and then my advanced students, we're all talking about the same. The biochem students were doing proteins just now. Same thing we're doing. Okay, just in a little bit more detail, angles and stuff like that. My colleague teaches them, not me. And they bond together. So you have the 8 or 9 of them, whatever it says, are ones that we have to acquire through diet. We can make all the rest ourselves. And so you have effectively an unlimited number of proteins or that you can make. The oddball thing is the term polypeptide and proteins. They are interchangeable. They are long chains of amino acids. Proteins are not more important. They're not more vital. There's a whole difference is number. If you have 100 or more amino acids, it's considered a protein. 99 or less, it's a polypeptide, unless it's just 2 or 3. Okay, that's it, right? And so typically what happens, how do we make proteins like insulin? It involves 2 of these, what we call polypeptide chains that come together. Hemoglobin, you will see, has 4. So when you add up all the amino acids, they're all chained together in some way, shape, or form. If it's 100 or more, it's now a protein. That's the whole thing. So that's really what sort of the background of all of this is. Now, structural proteins, I'm going to do at the end. The big thing to know about, and you all learned about, collagen, elastin, reticulin, and connective tissue. Collagen is the most abundant protein we have in our whole body. It is strong. It derives its strength from this. They are typically braided. Braiding is a way that we biologically, by twirling these linear molecules together, are able to create tensile strength. It's extraordinarily important to understand it. Moreover, these are all, with I can think of one exception, insoluble. So when you think of the interior of a cell or the exterior outside of the cell, those connective tissues, The collagen is holding cells together. It's strong. It's got what we call tensile strength. It can hold them. It can bend. It can stretch. However, it's not affected by the fact it's surrounded by a water environment we call extracellular fluid. On the other hand, there's one that's in the plasma that dissolves. You'll learn about it in blood. It's called fibrinogen. Blood is a connective tissue. That's the fiber. You got to have fibers to have connective tissue. The ground substance in blood is liquid and it has an abundance of cells. That fibrinogen is soluble. It is dissolved. The whole idea of the clotting cascade, how whatever makes your blood clot, and there's let's just say you tripped and fell and got a scrape, a cut in the kitchen, anything like that. Paper cut, trying to open an envelope, whatever. All that clotting cascade is accomplishing one task. It's changing that dissolved fibrinogen into its undissolved form called fibrin, which for all the world looks just like a fishnet that traps red blood cells and platelets and makes your blood clot. Shape, and shape. Recurrent theme today. So, enzymes. There's a whole section on enzymes. Enzymes really hold their shape. The area, and it doesn't, and this is just a generic view, one we talked about, making a disaccharide like sucrose from glucose and fructose. It doesn't always put two together. A lot of times it separates, and they can go in any direction. You could take 6 things and put them together. You could take something that's relatively complicated and pull it apart. Okay, so there's no limitation per se. Where they come together is called the active site. That's where everything occurs. That's a really important term clinically going forward. One of the ways that we treat patients with a variety of disorders is we begin to do what's called competitive inhibition. We put stuff in that will bond to the active site to stop a process. Okay, and that's a very, very important way that you will learn when you look at pharmacology. Many of you will do that. We talk about it in microbiology as treatments for diseases. HIV would be a very good example of using that. Other viral diseases, herpes viruses, we use these processes of competitive inhibition. So it's what I've told all the other classes today. I've been doing anatomy and physiology for more than 50 years. Half of that time clinically treating patients, half of that time attempting, with a modicum of success, trying to help educate future doctors and nurses. This is my way of understanding. I hope it works for you. I can't guarantee if this is clear to you or not. I hope it is. I hope it helps. If you need clarification, that's why I'm here. That's why the good Lord made email. Right? You can even talk to me in person. At least 3 of us. Joking. So nevertheless, if you see the suffix ASE, it's an enzyme. More on that coming up. Transportation, one of its roles. This is an example of 4, count them, 2 in this one brownish color, 2 in a little lighter color, polypeptide chains, less than 100 amino acids. Okay, and they have room spatially for an interesting compound called heme. The polypeptides are called globin. The heme is a little piece of iron, salt, surrounded by some organic material, and that happens to be a perfect vehicle to transport oxygen. And there's a whole bunch of those that you find in a red blood cell, to the tune of, are you ready? About 270,000 individual molecules like that in one teeny tiny red blood cell. So it just gives you an idea of how much oxygen carrying capability you have. By the way, you have like between 5 and 6 million, okay, red blood cells in a microliter. Wow. I mean, that's like 1,000th of a milliliter, and most of you know what a milliliter is. Okay. Gives you an idea how much oxygen carrying capability we require, why oxygen is so darn important. This is myosin. That's why it changes shape. This is the myosin head. When it's ready to fire, it's here. When it fires, it goes this way. You can see the arrows. It attaches to a molecule called actin, which is interesting because it's both soluble and insoluble. More on that down the road. We have a whole unit on how muscles contract and their physiology. And basically, it pulls the actins, which are attached to the borders of a subcellular unit called a sarcomere inside all of our skeletal muscles and basically pulls them together, shortening them. Muscles don't contract as much as they shorten. And when they shorten, depending on the shape of the muscle, and Dr. Robbins will show you some of the giant shapes of muscles that are there. Some are like fans, some have a belly in the middle. It's like when somebody flexes their muscle and they have a muscle that protrudes, it means it's narrow at the ends and wider in the belly. So when it contracts, it causes that to enlarge. So that's how you understand what kind of muscle it is. There's a whole way of naming them that I'm sure he will talk about. Unipennate, bipennate, multipennate. You're doing bones now, right? Muscles are next. Hey, there's only 206 bones. There's 620 muscles. I know. Annoying, isn't it? Anatomy. And perhaps most of them fall into this category, messengers. This is insulin to scale. This is an embedded protein in a membrane. Okay? Because of all those properties of the R groups, the ones that are lipid soluble can squeeze into a membrane. A biological membrane is a phospholipid bilayer. It can squeeze in between the lipids. It can even be held there by other forces that are there. Okay? On top of that, okay, it has part of it that's protruding outside the cell is water soluble. Part of it that's inside the cell is water soluble. That's the beauty of its shape. It folds and folds in such a way that the fat soluble things are generally on the interior. That's why it can do that. Now, insulin, a massive molecule, attaches itself to what we call a receptor outside and causes a change inside. So it's as though insulin, it's like we hear a knock on the door, a doorbell, insulin's ringing the doorbell, knocking on the door, and you'll do something inside, whether you go answer the door, whether you see it's a delivery, you have to sign for it, maybe you have anything along those lines. Maybe it's someone you could ignore, but you're going to have a response. And so here you're able, without entering the cell and altering it, able to be cause and effect inside the cell because you're changing the shape. What insulin does, because it's a shape change, such that it sends a whole series of reactions. It's not all about the sugar. Okay? Insulin causes your cells to accelerate metabolically. There's a whole chain reaction of things that occur with that particular molecule attaching. If you did not know it, insulin causes cells to replicate and uses sugar. That's why it's bringing in the sugar. Growth hormone, which we secrete all the time for repair, okay, basically does the same kind of a thing, except it uses fats for an energy source. And the two of them are usually, combat's the wrong term, when one's active, the other one is not. So we're either functioning to make more cells utilizing sugar as a source or utilizing fats as a source. And they both have problems attendant to them, like with anything else biologically when you do something or use something to excess. And lastly here, defensive antibodies, okay? You got a cold. This is called a naked virus, the common cold virus. Bacteria are a little bit different. They all have markers, okay, that identify them. Your immune system finds one that's causing trouble, that's been alerted. And you get to this in the immune system in AP2. It fashions an antibody, not the only way it fights viruses or bacteria, but it fashions, in this case, an antibody. This would be a cold, for instance, to that little spike of protein. It's about the shape on the outside of that that's protruding. You can't see it here. It's not depicted. It causes your body to produce this sort of a Y-shaped thing that has active sites designed to fit against the virus. It produces unbelievable masses of these that go through all your body. It's not magic. They don't go to your throat because you have a sore throat. They don't go to your nose because you have a runny nose. They go all over the body, and the more there are of them, the more likely they're going to collide. Why? Sometimes they produce temperature. It makes collisions occur faster. And so those antibodies will come into contact with, just by sheer weight of numbers, onto the virus or the bacteria, hold it, in this case, surround it. That tends to make them more visible to the immune system, and white blood cells come along and gobble them up. It deactivates them, stops them from doing damage, and the other white blood cells come along and go gobble it up. One white blood cell makes the antibodies, another white blood cell gobbles them. That's the way your immune system works in many, many ways. There's different ways as well. Just to give you an idea of the assorted powers of the proteins. So now we'll look at them. 20, a very strong, powerful bond called a peptide bond is how they attach to one another. Effectively, the acid on one side and the amine on another side of two adjacent, as you'll see in the illustration, bond together. Here's their drawing, infinitely better than my drawing. No, you do not have to know the chemical structure. Okay? But when we want to put two together, the amine group on one and the acid group on the other, and because we're putting them together, it causes water to be removed. If we were taking them apart, we would have to add water. So you'll see it here. And this, you can see it's a reversible reaction caused by, and since it's forming a peptide bond, the fancy word that I'm not going to ask you about, you might see it in digestive, is called a peptide ACE, A-S-E, with a suffix. And for instance, our bodies, when you had a burger for lunch or whatever you had for lunch, that protein, that effectively in our digestive system, the small intestine, the only thing we can absorb are single amino acids. So we have a series of peptide ACEs that break these down. And finally, in the lining of your small intestine, there are enzymes that break it down to its finite, to the smallest unit, which we can bring in. The same thing with like lactose being a sugar, too big to enter the cell. A single amino acid, small enough to enter the cell. The fancy name for this is counterintuitive. I don't want to ask you a question about it because it bothers me. I hate things that are that I would change the name if I had the power to do so. This is how a peptide bond forms. It takes the OH off the acid group, the hydrogen off the nitrogen. They form a bond together, that carbon to nitrogen bond. So COONH is a classic example of the peptide bond. They call this a dipeptide. Now, I don't know. That would mean two peptides, but that's wrong. It's right to me, but it means two amino acids. I didn't make the rule. It's a just because rule. It's just because some idiot. A tripeptide isn't three peptide bonds, it's three amino acids. That's why I'm not going to ask any questions about it. That would be rude. So, but if you see it down the road, I warned you. And that's the kind of material that sometimes anything that is inherently confusing to students who don't even want to hear about science and chemistry. Admit it, you're sitting back. There's a few of you who are curious about this, you're science nerds. The rest of you are going, I hate this. It's okay. Own it. It's you. It's all right. See, it's different things for different people. It's like some stuff, it injures me now. I used to hate, I was good at it because I had good memory, but I hate history classes. And now I love history. It's just something you might fall in love with it later. Give it a chance. So here are the structural levels. We start with a batting order. It's called the linear sequence. That's everything. That sequence determines whether it's going to be a structural protein, whether it's going to be an enzyme, whether it's going to be implanted in some membrane, whether it's going to be an antibody. That's all the basis of it. It's nothing more than a batting order like this. I just did 1 through 9 with a dash representing peptide bonds because it works. It looks like this. You can see now, if you look closely, and I was just listening to the biochemistry professor, my colleague, we teach the pre-med students for the MedCats, this very important admission test, biology. I do the life sciences, she does the biochemistry and the genetics and stuff like that. So what happens is every one of these has an NH. Remember, that makes hydrogen bonds. At the very least, any long chain of amino acids is going to hydrogen bond. And depending on what the R group is, there may be greater opportunities to make hydrogen bonds. That is the secondary structure. It can be one of two forms. It can either form, and this is why shape is everything. Think of your brain. Giant structure. How do we fit trillions of nerve cells in a small enclosure like your skull? We fold. We fan fold, we accordion pleat. Your stomach, normally unfilled, has a volume of about 2 ounces. It can stretch to a gallon. When you see it unfolded, it has ridges. Those ridges are folds, like accordion pleats. When you fill your stomach, if you had like a big dinner, or you went to a buffet or something like that, it's like this. When it closes, you see the ridges. They have a great name in anatomy. They're called rugae. Your bladder has the same thing because your bladder can expand from relatively empty to fill to maybe 1.5 liters or so when it's really excessively full down. Okay? That's fan folding. We see it everywhere. That long thing in your body called the intestine, the alimentary canal. Between your diaphragm and your anus, there's about, 20 feet, 25 feet. How do we get it all in there? Folds. Got the idea? What makes it fold? Those little dots, hydrogen bonds. It's all about the hydrogen bonds. There's always going to be an abundance of NH, OH, pH, and SH. The sulfur is a big player in this. Sulfur plays a role in a lot of hydrogen bonds, and it plays an important role in shape because those are hard bonds to break. Down the road, you'll hear about them. They're called disulfide bonds. Don't have to know anything about it now. It's something you might see a little bit more at micro, I guess, for many of you. That's the secondary structure. Hydrogen bonds. Test question. What causes the secondary structure? Hydrogen bonds. What's the primary structure? The amino acid sequence. Okay. And as well as what these are called beta pleated sheets, pardon me, it also can form helices. An alpha helix is right-handed. On Wednesday, when we look at DNA and RNA, you're going to see some right-handed. Here's another spiral. Right-handed spiral. I'll try to make sure I try the same way. Same deal here. Right-handed spiral. So it's a combination of these spirals and the pleats, which end up being shaped more or less like little ribbons that we see, and they come together to form the tertiary structure. That basically is how this polypeptide, in this case, will form. In this case, it's making a globular protein. Most of them are. Ones that have a lot of function, structural elements, always that great. And that's the role of the R groups. Acids and bases come together to neutralize each other. Positives are traded to other attractors. They take magnets. Water soluble, water soluble, lipid soluble, lipid soluble. That's how you make this. That is how you make this molecule together. Okay, that's the tertiary structure. A lot of the globular proteins are not only tertiary, but they have more than one. This is the same one, and there's two of them together. It's a fancy name called transthyretin. If you did not know it, thyroid hormone doesn't dissolve. It's fat soluble, but it travels in the bloodstream. It usually bonds to a protein. Remember, proteins have areas that are fat soluble and water soluble. The fat soluble attaches to the thyroid hormone. The water soluble gets dissolved in the bloodstream and it carries it. Nearly every cell in your body responds to thyroid hormone. It's extraordinarily important. When you get to endocrine, you'll learn about that. That's what it's about. The big problem, we don't do that at elimination, it's nothing to it. And we skip this. We're talking about globular proteins, ones that are soluble in water, not insoluble. We'll get to these in a moment. Compact, spherical, generally, water soluble, sensitive to environmental changes. The two things that affect it, anything that comes in there can alter it. They're designed, okay, to function around a pH of normal, which is around 7.4, okay, plus or minus 0.05, that's a 7.45. In general, the thing that impacts it, you see, we're much more prone to acidosis, acidic environment, than alkalosis. Okay, much diabetics, that's a real big problem, because they start to go, and this is the pH, the lower side means increased acidity. It's like a slippery slope. That's what this is a bell curve. Okay. Good news about it, by and large, unless it really slides all the way down, it's reversible. So we have somebody who's acidotic or alkalotic, if we address it quickly enough, the proteins will come back to where they're supposed to be. The one that's not is temperature, particularly hot temperature. Cooling something and indeed exposing it to prolonged cold, we have a certain degree of reversibility. If you pack things properly and you freeze them or refrigerate them, they have a pretty long shelf life. If you don't handle them properly in the freezer, they'll be a mess. It's not because of denaturing the proteins, changing them. It is because generally about mishandling freezing. It's the way to properly freeze things. So, with the temperature going up, the big problem is not cold. No, it's not a bell curve. Temperature starts like a bell curve. You hit 37 degrees Celsius, it's like falling off a cliff. You have a little window between the optimal temperature, we call that T optimal, and here, T lethal. That little range is about 5 degrees Celsius. That's 9 degrees. I mean, if you're 98.6 and you go up to 107.6, most likely you're not going to survive. That's irreversible. Irreversible changes. What happens with any alterations like this in pH or temperature, hopefully reversibly, but unfortunately sometimes irreversibly, proteins begin to lose their nature. They begin to change. When they lose their nature, they can't function. So take one that's a combination of all these things. One of the things insulin does is lets glucose in the cell. So when insulin attaches to a receptor, that receptor goes from looking like this, and all of a sudden looks like this. It changes its shape. It opens so the glucose can come through. When you take the insulin away, it closes. We have a lot of those. They're called gated channels. You get sick and tired of hearing about them. But different chemicals will cause different channels to open and close, changing the shape. Okay? So that's really important. I told you how antibodies work. There's so many of them, they latch onto. Once they latch onto, they can neutralize, they can tag it for white blood cells, they can basically do a variety of things to make it easier for the rest of your white blood cells, rest of your immune system to get you better. Molecular, I'll save hormones for a second. No, I beg your pardon. Let me do the hormones first. That was the insulin model. I skipped it out of there. As enzymes that are last. Molecular chaperones are just that. We have different kinds of proteins that escort other important molecules and put them in order. We're not going to get into the ins and outs about how proteins are made, but you have something that we'll talk about in passing called transfer RNA, and that's a particular molecule that's kind of like a chaperone. It latches on to one amino acid and pulls it over to the ribosome, and there's a batting order, and that batting order is back to this, similar to the batting order that comes from your DNA and your RNA is how you assemble proteins. That's a chaperone agent. There's others. And then lastly, enzymes, perhaps the most important. Because every biological reaction relies on enzymes. And enzymes of all the world, you could draw it like this. Big molecule, a little area that's elegantly designed to do something like that. It's all about shape. Effectively, the way they work is something called an induced fit. We used to think that the two would attach to each other. These logical keyboard. What happens as this, and you see different names for it, so get used to them now. I'll just call this, one people name it theater interchangeable. Substrate or a ligand. There's no difference. The thing that hooks onto to that part of the enzyme. That part of the enzyme where it attaches is called the active. What happens as that, either it's 1, 2, 6, whatever. There's one particle that's going to be split apart, or there's multiple particles that are put together. Effectively, if they get close to each other, both sides begin to change. So just the very proximity induces a change to permit it to fit. And that it really implies reversibility. As it gets farther away, it goes back to the way it was originally. That's the end. It was a little bit, a lock and key, to me, when I think of a lock and key, every so often a lock gets stuck very slowly. He does that in the water. That to me is problematic. That's what we kind of did away with that. What happens when things denature, I mean, if it's irreversible, that whole protein just loses shape entirely. But if it starts to denature, that little area, in the presence of alterations in temperature, pH, heavy metals in the system, there's a lot of things. All of a sudden, it's going to look like that. Shapes won't be compatible. You reverse it, it will resume its compatibility. That's the idea. So a lot of this has to do with it's a whole thing with shape. I'm a nut for it. If you haven't figured it out, you have been asleep through this, which would be hard. All this energy. I had 4 days of R&R. Nevertheless, that's what enzymes do. So here's denaturing. They unfold and they lose their shape. That whole big mass unfolds and it unfolds and it unfolds some more. But eventually, the one thing that doesn't go away, all that's left, it'll lose its quaternary structure, its tertiary structure, its secondary structure, and what will be left? Just the prime. Those peptide bonds are extremely strong. When you cook an egg, when you cook a piece of steak or chicken, okay, that's irreversible denaturing with temperature. It changes the shape. It's never coming back to the way it was. And the longer you do it, the more problems with the shape until it gets to the point where, tough, inedible. Irreversible changes are by the extremes, particularly the ones we see the most: increased acidity, lowering the pH, and increased temperature. Reversible most of the time, thankfully. That's the whole idea. When we get somebody in crisis or approaching crisis, you react quickly. Like in an emergency center, if we see somebody in ketoacidosis, you're moving. You're moving. They have a lot of acid in them. We start units of bicarb intravenously, just the way you would neutralize something. You pick something that's mildly alkaline and try to bring it back. That's what your gut does. It takes acid and adds bicarb to it. Structural proteins are different. They're fibrous. They braid. So if you have helices like these, okay, an individual by itself doesn't have a lot of strength. But when you braid them together, it does. Okay? Try it yourselves. Just try to braid 3 things together. That's the structural strength from fibrous protein. They're strands, but frequently they're braided. They are water insoluble, with the exception of the fibrinogen I can think of offhand. The actin we'll get to, it's a little different. Interestingly enough, when we get to it, you have things in your body that we learn in the next chapter called microfilaments and microtubules. They can form and become insoluble, and they can break apart and become soluble. Very interesting. It's sort of like internal roadways and passageways. And they are being built and broken down and built and broken down continuously in our cells. All the time. The inside of a cell is not static. You got to stop looking at that illustration on the board with the nucleus and the organelles. It's all moving around. All the time. So examples, you got these before: keratin, elastin, collagen. We'll get to the contractile ones right away. Here's the idea of denaturing once again. Fibrous proteins, if anything, those changes make them tougher. And that's not necessarily a good thing. So it creates an environment where it makes the internal area almost impossible to get anything in there. It's almost like a boundary, if you want to think of it that way. That's there. What enzymes do, here's another test question. By definition, they reduce the energy of activation. Just like there's enzymes for everything that goes on, in order to make something happen biologically, it requires a certain amount of energy input. So that the energy of activation, even if it's just to make 2 things, more likely to come together. So we know we have to put energy in. The question is, do we end up creating bonds and storing energy, or do we release energy and break apart bonds? That classifies anabolic and catabolic. That's all. In the form of metabolism. They're all reactions. All of them require energy of activation. The reality is that without the energy of activation, things would go very slowly in your body. So you would not be processing what I'm saying to you. I would not be able to speak to you that rapidly. I couldn't be able to do other things with a mild degree of coordination. Okay, all of that's part of it. Did I? You know what happens if I took another piece of paper towel, put it over here, assuming nobody put a thing on it, do not touch. If I came back tomorrow, assuming nobody touched it, would it look the same? Yeah. Next week? Yeah. Next year? 100 years? Do you ever go to find an old library book that has kind of yellowing and you opened it up, the pages were full? It's paper. It takes time for the moisture in the air to be able to break down that paper enzymatically. Okay? No activation energy. But if you ever go there and you're one of those age-old books in the old, I don't know if our library has these old stacks. Big university libraries have enormous areas that you're not even allowed to go into. You have to, a lot of them are climate controlled for old books. The Vatican has a whole climate controlled area for all sorts of old religious documents and all sorts of scrolls and stuff like that. Very interesting because it's so fragile. Not here. So we have to make things happen rapidly. And as you can see, millions of reactions per minute. The whole enzyme, I don't care about the name, holoenzyme, stands whole enzyme. Apoenzyme means the protein portion. Okay. The two things I care about are here. They are cofactors and coenzymes. They are also essential. By definition, and nearly every enzyme requires them. The cofactors we call minerals. So usually in order for that to form, maybe it's 2 or 3 chains, sometimes in the middle of it, like hemoglobin. With iron, insulin has zinc. So somewhere, let's say this had some zinc. What happens here, that little portion, so minerals are inorganic, maybe this little area is made by a vitamin. That's a cofactor. By definition, that's important. So again, vitamins and minerals, effectively, their major role is to make enzymes work better. So we call them cofactors for minerals and coenzymes for vitamins. Yeah, maybe there's some exception. Folic acid actually is in the B vitamin family. So I wouldn't go crazy about the exceptions. So the substrate, term ACE is typically, basically peptide bonds would be peptide aces. Oxidase doesn't imply oxygen, it's about redox reactions. So oxidizing means you're losing electron. Why is there oxygen? That's the parking place we see in biology. And here's lowering the activation energy, priming it, whatever you want. I mean, all the steps I really don't care about. This is a really good illustration. It sums it up right away. Normally, we require a lot of, how long would it take for that paper to start to break down? A very long time. But in a bio, we don't have that time. So the activation energy speeds it up. Temperature speeds it up. It's all about collisions. Okay. The activation energy is like that. Basically, it's imparting a certain amount of energy, and if you want to translate that to motion, the more likely things are going to collide, and then the size and the shape will complete the reaction. And they're just showing you that. There's this complex forming the bond, releasing water. So now you've got that peptide, dipeptide. Any way you want to represent it, just like that. It's fine. You could be putting them together, taking it apart. It'd be the same enzyme. And that is everything. So really, we'll look at the coding and how how these magical proteins are made without going crazy about it, but enough so you know the basic roles of RNA and DNA. We're not going to talk about mutations or things like that at this point. Again, that's more If you really, there's a big unit on for micro, and most of you are in the taking micro at some juncture. So thank you very much. See you by Wednesday. Oh, Joe, stop. In the name of presenting, stop recording.